Our first speaker is from the Linz ELLIS unit. It's Guenter Klambauer. He is also with Johannes Kepler University in Linz and is leading a research group on AI for drug discovery. And it's your floor, Guenter.
Thanks, Sepp, for the introduction. So I'll try to share my screen. Okay, so I would like to thank, of course, the organizing committee for giving me the opportunity to speak here. I guess, as Sepp has already said, so we are an ELLIS unit in Linz, located in Linz, and I'm leading a group there on drug discovery projects. I call it AI and Life Science Group with a lot of people. And we had in the past and in the present, we always worked together with pharmaceutical. So only for the there will be about in the molecular scale. So we've heard a lot of things about patient scale, like tracking, also maybe microscopic or patient level or patient scale, things like healthcare management. And the speaker before me have already introduced genome sequencing, which would be targeting or doing relating ML and using ML and AI methods in the molecular scale. So there are multiple ways how you would approach this. So on the left-hand side, you see a schematic representation of the coronavirus. So these purple things and so on, these are proteins, structural proteins, for example, that the virus has. And in a drug discovery approach, you would try to design a small molecule, something like that, to inhibit or to hamper the virus, for example, to break some of its proteins or to inhibit some of the machinery that it needs to replicate and to spread. So there are multiple ways. So it has this coronavirus is a relatively simple thing with only a handful of proteins, but you still have a lot of ways how you could approach this to basically hamper the virus in a drug discovery approach. Or you could also think of antibodies. I will not focus this talk too much about this, but also antibodies could be designed to inhibit the protein, and that's basically what our body does. So how do we get to the computer-aided drug discovery then? A lot of work is currently in such a drug discovery project and process. A lot of work is done already on the computer. You see that there are a number of things listed. I will only detail two things, and very quickly I will talk about compound design, which can be done by machine learning, and also virtual screening. So machine learning can influence all these areas. I will show a bit in these two areas what it can be done, and then I will show an example project, what we have done. Okay, so compound design, there has been lately a lot of work since 2016 or so on using machine learning methods to generate molecules. So one of the first was an LSTM using generating compounds, a string representation of compounds, but there is now variational autoencoders generating molecules. There are GANs generating molecules, also reinforcement learning in combination with RNNs, for example, and even hybrid approaches. So overall, I think there are around 90 to 100 publications suggesting molecule generator with machine learning techniques. And these are very valuable strategies. However, I want to, so they can be, they can be a bit slow in the current crisis, because these molecules, they have to be still synthesized and so on. So it could be a bit faster if one does virtual screening, which is the second thing. So virtual screening is basically using a computer, using a model, machine learning model, to screen through a large database of molecules that are already available. So typically there are a lot of large databases around of molecules that are quickly available and can be purchased and tested in the range of millions or billions even. And there are two principled approaches, like ligand-based and structure-based approaches. Ligand-based approaches, they revolve around the ligand, which is the small molecule here that we want to design, and structure-based approaches. So these are computational techniques that basically use the 3D shape of the 3D coordinates, the 3D shape of the protein, and try to dock a small molecule, try to fit the small molecule in. Involves typically molecular dynamics situations. And of course, also here there are hybrid approaches. Again, I will focus here on the ligand-based thing. Of course, also in the structure-based area, machine learning can play a role. But here it's very clear how machine learning can be used in a ligand-based, because you typically have labeled molecules that can bind to a protein or that can inhibit the protein, and small molecules that do not have this property, and you can build supervised models, then perform virtual screening and select some compounds. Overall, this is a typical drug discovery pipeline. So it's a very famous figure. It can take very long, so it can take even up to a couple of years to finally develop a compound. So here you see a very long time scale. Of course, this can be much faster, but it's very typical that one starts with a large compound library with millions of compounds, then uses a virtual screening approach to select a compound library in the range of tens of thousands that are then slowly reduced to a couple of favorable candidates that go into clinical trials, which also take a long time. And then if you're lucky, you get one approved drug. So now with the current corona crisis, we have to speed this process up a lot, and here machine learning can help a lot, because if you select very good molecules at the beginning, you can proceed faster, and you have a lower chance that accurate machine learning models can already predict side effects or low efficacy and so on. So how does this present from a machine learning perspective? From the machine learning perspective, it's quite simple. So I'm presenting also a simplified view here. So your input are molecules, and you have labeled data, actives and inactives in the simple case, and you build a model. But what is the typical case? That you do not want to predict or know the activity of a molecule on a certain biological effect, or whether binding to a single protein or not, but typically you want multiple effects at once. And here you can, for example, use multitask learning to have in a simple neural network with multiple output units that model multiple biological effects of a small molecule at once. And it's very typical that such label matrices like this here have a large percentage of missing labels. So this is very natural here that we don't have a lot of knowledge. This also represents our knowledge about small molecules, that the large part of the effects of small molecules we do not know. Also, from the machine learning perspective, molecules are very interesting from the representation view. So there are different ways how you can represent a small molecule. Of course, you can extract some features and represent it as a vector. The community calls this so-called fingerprint features. There is a string representation that I quickly already announced called the smiles representation, which goes neatly together with the RNNs. Of course, very natural, and we all learned it in school. It's a graph representation. Here, the graph neural networks could work, and there are also other representation, like with sets of fragments, and also representing this as a point cloud and using some point-based neural networks. So all of these representations can go with some machine learning techniques. Some years back, it was already shown that deep neural networks outperform other techniques at this task of predicting a drug target. So given a molecule, predict to which proteins it will bind. The gap was not as huge as for image recognition, but still it improved in all of the last years with attention mechanisms, and on this could be further improved. And commercial software at that point of time was far off at predicting drug targets. In another example here, for example, also toxic effects can be predicted quite well using a deep neural network with in a multitask setting, predicting multiple neural net toxic effects at once. So here would be toxic effects, and here would be the average thing, and deep neural network can do that very well. It's even the case that for some tasks, for some proteins, for some of the drug targets, the predictive quality is so high that a wet lab procedure can be replaced. So typically, you also are from partners from pharmaceutical companies say, if you reach an area in the ROC curve larger than 90 with a model, they consider this a virtual assay and put thinking about replacing a wet lab testing procedure with a computational technique or machine learning model in this case. Why can it work so well? Because there are typically large databases for learning. So for example, the PubChem database offers 270 million triplets. A triplet means a molecule measured for a particular target, a drug target, for example, a protein. Can this molecule inhibit this protein? And you have an activity value, which can be binary, but also regression tasks, of course, make sense. But overall, so PubChem and Campbell offer these activity data. DrugBank is a data set of marketed drugs, and there is a Sync database, which is a large, contains a large number of molecules. So assume you have a trained neural network, then those which select the molecules that can use potential toxic effects. Am I still hearable? Okay.
You were gone, but now we hear you again.
Switch off the video for now.
Okay. And I'm showing the result of such a study. So we applied this concept of virtual screening recently to coronavirus inhibitors. We were a bit lucky that we had a pre-trained network called that was trained on a large number of biological effects. So that data set comprising of Campbell, PubChem, and Sync. And so this network, the architecture is so-called SMILES LSTM. So it works on the SMILES representation and employs an LSTM, predicts multiple output at once in a multitask setting. And of these 600 different effects that it predicts, 4 were related to coronavirus. So there was some data present in public databases about the coronavirus, which is the old coronavirus SARS-CoV-1, and concretely on 2 very important proteins that the coronavirus needs for replication, so-called proteases. And we use this predictions to screen a database. And the good thing is that these 2 proteins are relatively conserved between SARS coronavirus 1 and 2. So we could relatively fast perform a virtual screening of the Sync database and report a list of molecules that could be potential inhibitors for the SARS coronavirus. We provided this list of the 30,000 top-ranked molecules via GitHub. I will share, the link will be at the end of the presentation. And so these molecules that I'm showing here could be potential inhibitors of the coronavirus and could be tested. The good thing about the Sync database is that it directly, so you can click on each molecule and find a vendor and relatively easily get this molecule purchased and tested. We also screened with this model, of course, DrugBank, and we got a couple of we got a ranking of molecules that are already on the market, so marketed drugs that would have the advantage that you can test these drugs faster in clinical phases because they're already on the market, so there could be a faster way to get into drugs. These results on DrugBank can be requested from us, and we would share it for scientific purposes. My last point, I'm almost finished. So there will be a similar challenge announced very soon from a European initiative, so the JEDI initiative. and it's going to be the billion molecules against COVID-19 challenge. It's very similar to what I've shown. So teams like machine learners, groups with ours are asked to provide lists of molecules, asked to screen large databases for potential molecules that can inhibit this virus. They should have at least should be screened for at least 5 drug targets. So I showed results on 2 drug targets, and in 2 follow-up stages, these predictions from the different methods and teams will be tested experimentally and validated. This will be announced very soon. It's very short, so running only a couple of weeks, open to all teams in the world, and the prize money is over 2 million euro distributed across 3 challenges. So yeah, there's already some information. Please stay tuned. The challenge starts anytime soon. Okay, so just a quick wrap up. So I've shown how machine learning can help in this corona crisis in the area of drug discovery. Machine learning methods are already employed, but there's still a lot of room to improve. Large data sets would be available, and machine learning can especially help because these are quite fast methods. And I've shown a screening of a very simple model, pre-trained model, a screening of the Sync database and the drug bank database. So all of this information and also the slides will be on the Git. So the GitHub is already on. There is already also the small technical report on what we did, and of course the list of the molecules. And we linked a lot of database, and also of course this the data set that we can be used for training of these 4 essays related to the SARS coronavirus is uploaded there. Yes, so that's it from my side.